Welcome to Health Professional Radio. I'm your host, Neil Howard. Thank you for joining us for another segment. We're going to be speaking with Dr. Kleanthis Xanthopoulos from Shoreline Biosciences to talk about the development of genetically engineered induced pluripotent stem cells, or iPSC, derived allogeneic natural killer, or NK, and macrophage products. Welcome to Health Professional Radio, Dr. Xanthopoulos. Thank you for joining.
Thank you very much, Neil. I appreciate the invitation.
Well, give us a bit of your professional background, if you would, and talk about your role at Shoreline Biosciences.
I have a scientific background with a PhD in genetics and molecular biology, and then I was a faculty professor at the Karolinska Institute in Sweden before I moved to the United States in the early 90s to join an exciting super project in mapping the human genome. And from there, I moved to California, where I joined my first biotech company, Aurora Biosciences. That company was eventually sold to Vertex, and 4 companies later, as a founding CEO, I'm extremely excited to lead Shoreline, that I am the CEO and chairman of the company as well as a co-founder.
Exactly what is Shoreline Biosciences involved in? I know that I mentioned that we're going to talk about some of your products, your iPSC derived allogeneic natural killer and macrophage products. Explain what these products are.
Yeah, about a decade ago, some very elegant work originating from NIH and ultimately coming to the industry through companies like Kite and Juno demonstrated some wonderful results in hematological tumors based on the concept of cell therapy. The idea was we're going to take the cells of the patient, manipulate them ex vivo outside of the patient in our laboratory, arm them with specific antigens, and then reinfuse them into the patient. That is the autologous CAR T therapies. CAR stands for chimeric antigen receptor, which, if you like, is the guiding missile that we put into these T cells. And the results were wonderful. We saw complete responses in some very significant hematological tumors in the excess of 60, 70%. And now, 10 years later, we have half a dozen products in the allogeneic CAR T, in the autologous CAR T space by several companies. The problem has been that these are highly effective, but they are expensive to manipulate. It takes about a month or more, sometimes more, to manipulate these cells. Unfortunately, for these severely ill patients, they don't have the time. They don't have a month or two. And in addition, we saw some safety signals in a small percent, but nevertheless recordable and repeatable percent of patients that developed some very significant adverse effects, such as cytokine release syndrome. We saw graft versus host, and we saw some deaths associated with the treatments. Despite that, these therapies continue to advance the frontiers of cell therapies. However, several years ago, other effector cells began to also appear to have an impact in the clinic. And those are specifically natural killer cells, and we're very soon going to find out also how macrophages behave if we manipulate in the same way. So, as you can imagine, Neil, we don't respond to a tumor or an invader, which that might be a virus or a bacterium. We don't respond with only one single cell, like we mount both an innate and an adaptive immunity. So there are multiple aspects that one can manipulate. The advantages of NK cells are they appear to be shorter lived in the patient, and as a result, they don't have these side effects, yet they appear to have a very strong and good efficacy. So our idea is that we can use NK cells and macrophages to complement all these cell therapies that are out there on an autologous basis, but do it in a way that is affordable. It is of the cells, i.e. that's why we call them allogeneic. We can literally have these cells in the freezer and give them to the patient as needed instantly. We don't have to wait for a month or two. And we think we're going to have a safe, affordable, and very and therapies that are very effective.
Now, you genetically modify these cells. That's correct, right?
Correct. We start from a cell source that is called pluripotent stem cell. And as the word indicates, this is a cell that can be depending on the different conditions that we grow these cells, we can differentiate them in different hematopoietic lineages. So we can take them towards the direction of a natural killer cell and create mature natural killer cells from pluripotent stem cells, or we use different conditions and we can drive them towards monocytes, macrophages.
Is there a limit to the extent they can be targeted?
Theoretically, no. What you have to make sure is that after every genetic manipulation you introduce, whether it's eliminating a gene because you make these cells metabolically better and more effective, or you introduce a construct that targets them to a specific antigen for a specific tumor, those are all steps that theoretically are unlimited. However, after each step, one has to make sure that you have 100% achieved the original goal before you take the next step. And this is, again, why starting with pluripotent stem cells can be a huge advantage. There are other companies trying to do the same thing with NK cells using umbilical blood cord or peripheral blood derived NK cells. We think the pluripotent stem cells, for a variety of reasons, mostly technically superior ways of doing this, are the way to go.
During the summer, Shoreline announced a couple of collaborations, actually 3 of them. I'm hoping that you maybe can walk us through these collaborations and tell us what they mean for Shoreline.
Yes, we view those as transformative for Shoreline and validating for the field. So we established 2 major collaborations and a third minor that I will describe very shortly. Those were with companies like Beijing and Kite. These are some very innovative companies that share our vision of creating allogeneic off-the-shelf affordable and safe medicines. And they have done their homework. They looked at the landscape and identify our capabilities, technologies as being superior. We enter into these 2 collaborations worth over $4 billion and with very significant over $100 million in upfront payments that enable us now to fast forward integrate our own capabilities, because both Beijing and Kite are very much involved in how we build our programs and how we take them to the clinic together. They are truly transformative because now it's the coordinated efforts of 3 different companies addressing some very serious oncology indications, both for hematological and solid tumors. The third one was with manufacturing capabilities enhanced by a collaboration we established with the Advanced Cell Therapy Laboratory, which was originally created here at the University of California in San Diego. And that allows us then to create a clinical material that we can test as we're entering into phase 1.
Well, I appreciate you joining us here on the program. If you could give us some information where our listeners can go online and learn more about Shoreline, it'd be appreciated.
Yeah, I'd be happy to do so. Please visit our website at shorelinebio.com. We also have a long presence at LinkedIn under Shoreline Bio, as well as Twitter and other social media, where you can find a lot about background about our company and also videos about how these cells work in humans and what our ultimate goals are, which is to help these severely ill patients.
Dr. Xanthopoulos, thank you so much for joining us on the program.
I very much appreciate it. Thank you.
You've been listening to Health Professional Radio. I'm your host, Neil Howard, in conversation with Dr. Kleanthis Xanthopoulos. Audio copies of this program are available at hpr.fm and healthprofessionalradio.com.au. You can also subscribe to the podcast on iTunes, listen in, download at SoundCloud, and be sure and subscribe to our YouTube channel at youtube.com/Health Professional Radio.